Hello and welcome to Lecture 11, Part 2 of Life in the Universe. In this video, we will continue with Unit D, looking at the history of life on Earth. And in particular, we're going to be taking a look at some ideas about the origin of life. How do you get from molecules to cells? So first off, if we're going to get life going on a planet or really anywhere else, we're going to need some raw materials, basically simple organic molecules that we can then put together in these more complex ones that we've talked about in the previous unit. And people working on this problem do try and be pretty careful that there's actually evidence that these molecules are in fact available. And everything we've really found indicates that, yeah, these simple organic molecules are likely to be reasonably common. Now, we also need to know something about the Earth's early conditions. And one thing that's clear is that Earth's early atmosphere was quite different from today's. Our initial atmosphere would have been probably mostly carbon dioxide with nitrogen, water vapor, probably some methane and ammonia. And so incidentally, CH4, that's methane, NH3 is ammonia. One big difference is that there had been very little molecular oxygen in the original atmosphere, and that's for reasons that we will get to as the course goes on. Now, various laboratory simulations have been done with plausible earlier Earth conditions and have demonstrated that as long as you do have those basic simple organic molecules, you have some source of energy, sunlight, geothermal energy, lightning, whatever, that you can produce kind of a next level up in organic molecules, amino acids, sugars, lipids, purines, pyrimidines, remember those are bases of RNA and DNA, and ATP, adenosine triphosphate. And once you have those, and you've got the building blocks for proteins, cell membranes, and RNA and DNA. And it is known also that some of these exist in extraterrestrial circumstances as well, and in particular in primitive meteorites, indicating that they would have been widely available in the early solar system. Now, we talked about this a little bit in the last unit, but once you get amino acids and lipids in water, then they can spontaneously form enclosed droplets, probably as analogous to the very earliest cell membranes. I will point out on the one on the left, which shows some little amino acid membranes, that where it says down here that they're not alive, but they have many lifelike properties. What that means is that they can grow by adding to themselves from other amino acids in the environment, and in a sense reproduce. They grow to be big enough, they tend to split into smaller pieces that can continue to grow. So not far enough along where we want to consider it alive, certainly, but obviously a step in the right direction. Now, the general consensus, or certainly the majority opinion, it seems among biologists working on the origin of life on Earth, is that RNA probably came before DNA. You could have had something else before RNA, but the RNA seems to have come first relative to DNA. And so you see the RNA world term sometimes used. Abiotic, remember that means formation without biology. And anyway, another thing we've demonstrated in laboratory experiments is that you can get abiotic formation of little bits of RNA that self-replicate, because that's largely what nucleic acids do in presence of other nucleotides. And even you can drive evolution of them if the environment will prefer a particular sequence of bases on your abiotic RNA strands, then those will self-replicate more efficiently than others. Now, of course, you got to get those things inside the membranes if you're going to take a useful step towards making a cell. And so experiments that have done where you abiotically produce RNA along with lipids so that you can form these membranes, sometimes that, as we see here in the center, you get that some of that RNA enclosed within some of those membranes. And so we wouldn't call this alive, I don't think, at least not unambiguously. But you do have what we might consider a pre-cell here, in that you have genetic material in an enclosed membrane. Now, again, a reminder that if you've got other RNA nucleotides around, RNA will act as a catalyst for reproduction of itself. And we've also seen that RNA is involved in stringing together amino acids to make proteins, or polypeptides more simply. So probably what happened, in addition to catalyzing its own reproduction, some of this early RNA was able to put together some probably very small proteins compared to modern ones, but you hit on one that will catalyze RNA replication, and you get the RNA reproducing even faster. Now, if this is just happening out in the general soup of things, then RNA strand that produces a little protein that will help RNA reproduce, that's not an advantage for that strand of RNA. So it'll help all the RNA reproduce. But once you, wasn't where I thought it was, once you get the some of that RNA that makes those little early proteins that can catalyze or serve as serve as an enzyme for RNA replication, enclosed in a membrane, that only helps the RNA in that membrane and not other bits of RNA. And that's one of the importances of being enclosed in a membrane. And so this is still a pre-cell, but it's the only thing it's missing from becoming a full cell is metabolism, and then we'd consider it alive. Now, I've got a bit more to do, but here's another one of these things where I'm going to put the attendance secret code a little early, just to make sure that people aren't skipping to the end. And so it turns out, I believe there are only 2 known statues of Charles Dickens in the world, which might be surprising since he's probably the most popular, or tied for the most popular English author of the 19th century. But apparently he didn't want a statue made of him. So in general, people have followed those wishes. But there are apparently 2, at least 2 that I know of. There's one in Australia, and there's this one, which is in Philadelphia. And it's in a park in somewhere in West Philadelphia. And it's him and one of the characters from one of his books. And so anyway, Charles Dickens statue is our code. All right, now, what about metabolism? Well, metabolism, the chemical reactions involved that, don't necessarily have to be connected to genetics. So the origin could have been completely separate, and maybe even before genetics. And if you think about it, that efficient metabolic reactions are going to be more likely to happen than inefficient ones. So you could have some selection on natural selection acting on the more efficient ones, even before there were genes. Because I guess you don't really need genes for Darwinian evolution necessarily, because what you need is the information on how to do something like run a metabolic reaction, and the reaction itself has the information on how to do it. But in any event, whichever came first, once you get enclosed within the same membrane, metabolism and genes, then you've got something that we would clearly call this is a cell. A very primitive cell maybe, but this is a living cell. And at that point, it's unambiguous that we'd have life. Like I said, we think RNA probably came before DNA, but once you can make DNA, it's a bit more stable than the RNA, primarily because it does a double helix better than RNA does. And so that's probably being more efficient at faithfully storing genetic information, is probably why DNA has largely supplanted RNA, at least as the storage for the genetic information, even if RNA still does a lot in terms of actually exercising that information and making proteins. Now, where did the first life on Earth occur? Well, one popular model, and there are other options, but On the ocean floor, you have this basically geothermal vents. So basically low-level volcanic activity where you have heat and so therefore energy and a wide variety of minerals. So you got a lot of chemicals available for doing chemical reactions on. On the seafloor today, these geothermal events are actually the anchors of these little ecosystems with bacteria and archaea uh, living off of the chemicals coming out of the vents and then other things feeding off of them. And that would be that in our at least two of our three possible dimensions we talked about on how to classify metabolism. The first organisms in this case would have been chemoautotrophs. There are other possibilities, but this is a popular one. We'll see there's probably some evidence for that, which we'll get to in the next video. And anyway, there's a lot of debate about the details, but here's a plausible description of the important steps involved. So we start off with the simple organics, then to make the slightly more complicated ones to serve as building blocks for the bigger ones, indicated here. Presumably there was not an actual chemical flask sitting on the Earth 4.5 billion years ago. That's there just to represent the experiments we do to try and reproduce these conditions. Anyway, along the line we get early RNA molecules, which become self-replicating and make some simple proteins like that right there. Some of this stuff gets enclosed within membranes. You add metabolism and you have the first true cells, which would have probably looked a lot like a bacterium. So let's call it a prokaryote of some sort, but with an RNA instead of a DNA genome. And then somewhat later you get the DNA genome, and then you've really got life as we have it today. Now, when did life originate? So here's our great moments in the history of Earth thing before. Well, that's a good question. We've said it would be difficult to survive out of the heavy bombardment, but it may have originated during or before that time. So let's see here, 3.8 to 4.3 billion years ago. So we see there is some evidence in that. So somewhere in that region is probably around the time when life originated. So actually maybe a little, 3.8 maybe even comes up a little bit higher. Anyway, on this diagram, the origin of life is probably about as shown as late as it plausibly could have been given the evidence, and may well have been a bit earlier. Okay, so that's it for this video. There's one more to do for this lecture, which after Zoom has converted this one into the proper format, and I checked to make sure that it seems to be okay, I will record that one, and that will conclude our lecture 11. See you in part 3.